In this video, we would like to introduce the DOHaD research we started in 2014 based on endocrinology and perinatal neonatal medicine. Undernutrition during pregnancy is one of the causes of low birth weight, and this increases the risk of developing diseases later in life. We are researching these mechanisms. Also, we're examining the effects of nutritional interventions in order to develop preemptive medicine for low birth weight babies. To do this, we created a low birth weight model rat from embryonic malnutrition. DOHaD means developmental origins of health and disease. This theory suggests that an undesirable environment during the embryonic stage to early childhood increases the risk of developing chronic diseases after adulthood. For example, when a low birth weight infant grows up in an environment rich in nutrients, there is a mismatch between the constitution of the child and the environment, and this is thought to lead to the risk of developing diseases in the future. It is hoped that interventions during pregnancy will reduce the effects of lifelong metabolic and psychiatric disorders in children. Various epidemiological studies form the basis of the idea of DOHaD theory, some of which are believed to have been derived from the analysis of the famine quasi-experiments shown in this slide. In particular, comparing the results of the Leningrad famine and the Dutch famine provided an opportunity to understand the thrifty phenotype and the increase in the risk of diseases. Let's look at that now. The siege of Leningrad began in 1941 with the invasion of Operation Barbarossa by Nazi Germany and continued until 1944 with 670,000 deaths officially reported and more than 1 million deaths unofficially announced by the Soviet government. There is no record of accurate food intake during the Leningrad famine, but from Tanya's diary, there is a description that the daily food intake was only about 300 to 500 grams of bread, due to the blockade, which lasted about 900 days. On the other hand, the Dutch famine had a short but hard food blockade from the fall of 1944 to the spring of 1945, and the daily food intake dropped to 600 kilocalories. This quasi-experiment is informative because all food was recorded in the Dutch famine. More than 75 years after the war ended, many follow-up studies of children from pregnant mothers were summarized in the Dutch Hunger Winter Study. This study found that children from mothers who experienced famine in the early stages of pregnancy had these conditions: abnormal glucose and lipid metabolism, obesity, abnormal blood coagulation, cardiovascular disease, breast cancer, stress responses, cognitive behavioral disorders, as well as depression. Children from mothers who were in the second trimester were more likely to develop glucose metabolic disorders, lung diseases, and renal dysfunction. And children from mothers who experienced famine in the third trimester were more likely to develop glucose metabolic disorders. On the other hand, the victims of the famine of Leningrad were followed up in 2009 and reported in this paper here. There was no difference in BMI and obesity, blood pressure, lipid metabolism, glucose tolerance, and risk of developing cardiovascular disease. However, they had short stature and tended to be underweight. We learned two things from this comparison. The first is that children get smaller when exposed to undernutrition during pregnancy. From this, the trade-off theory was developed. The idea is that if the mother does not provide sufficient nutrition, the fetus will change its metabolic and endocrine system to become smaller in order to protect the weight of the brain. The second is the idea of the thrifty phenotype. This was proposed by Hales and Barker as shown in this slide. Malnutrition during the embryonic period causes changes in metabolic and endocrine systems, which can be predictive adaptive responses that work efficiently in a poor nutritional environment even after birth. However, if the postnatal environment is a rich nutritional environment, a mismatch between the phenotype and the environment will occur, and this mismatch can increase the risk of developing metabolic disorders. The idea of this mismatch has evolved into the current DOHaD theory, and it's now clear that it's involved in the development of various non-communicable diseases, including not only metabolic, but also neuropsychiatric diseases. This slide shows the changes in the average birth weight over time and low birth weight infant birth rate over time in Japan. As shown by the blue and orange lines, the average birth weight in Japan has decreased since 1980 and remains lower still now. In addition, the birth rate of low birth weight infants, which is shown in the green and purple lines, has increased since 1980 and is higher now. Combined with the DOHaD theory, low birth weight infants, who are currently about 10% of newborns, are at a high risk of developing non-communicable diseases. It is believed that one of the causes of the increase in low birth weight infants is that pregnant women are lean. This data is a bit old, but even pregnant women who were lean before pregnancy gained 1 kilogram less during pregnancy in 1999 than in 1988. One of the causes of increased leanness in pregnant women and younger women is the lack of calorie intake. Calorie intake is declining year by year, possibly due to overexposure to advertising of low carbohydrate diets. For these reasons, there is a need to establish preemptive medical care aimed at early intervention as soon as possible. However, experimental verification of the DOHaD theory has time and ethical problems, so we decided to establish an animal model. The thrifty phenotype seems to have a higher predisposition to acquire non-communicable diseases, but the details of the mechanisms underlying this tendency are still unclear. Therefore, we generated a modern Japanese-style thrifty phenotype model rat that was fed a low-carbohydrate, calorie-restricted diet during the whole stage of pregnancy. By suppressing the calorie intake to 60% of the normal control rats, it was possible to suppress the weight gain during pregnancy of the mother rat, while the gestational age was the same as control rats. There was no difference in birth number and survival rate between the two types of rats. We obtained low birth weight offspring from the calorie-restricted mother rats. These low birth weight rats got milk from their mothers, who were fed the control diet after birth. In spite of this, we found that there were short body length and low body weight rats that failed to catch up with normal growth at the 21st day, which is the weaning day. In these graphs, this is the normal birth weight, and this is the low birth weight. And this means rats that caught up with normal body weight, and this means rats that failed to catch up with normal body weight. The growth hormone IGF-1 axis was examined to clarify the mechanism of acquiring a small physique by trade-off. The blood GH concentration on weaning day did not show any statistical difference. On the other hand, the blood IGF-1 concentration was lower than the control rats, and the GH receptor expression level in the liver was significantly decreased. We investigated the DNA methylation of the GH receptor code region, but no difference was found within the range we examined. So, we considered the involvement of microRNA as a mechanism for the decrease in GH receptor expression. From the database search, we found 5 microRNAs that have a sequence to bind to the 3' UTR of the GH receptor gene. In this comparison, we found that the expression of MIR322 was upregulated only in low birth weight rats that failed to catch up with growth. We also found that MIR322 was contained in blood exosomes and that the exosomal MIR322 was also increased in low birth weight rats that failed to catch up with growth. We also found that the expression level of MIR322 in the liver showed a significant negative correlation with body length and a significant negative correlation with GH receptor expression. Overexpression of MIR322 reduced GH receptor expression, which indicates that MIR322 is a factor that suppresses GH receptor expression. Since MIR322 is expressed in various animal species, the binding sequence of MIR322 is also conserved in the GH receptor 3' untranslated region. This may be a biologically important regulatory system and may play an important role in the trade-off in low birth weight due to embryonic malnutrition. Next, we noticed that our low birth weight rats were restless and naughty. When the restraint stress by a wire mesh was applied, we found that the corticosterone concentration in the blood was maintained at a high level, so we plan to investigate this mechanism. We previously reported that the negative feedback regulation of glucocorticoids includes a corticotropin releasing factor receptor expression inhibitory system by MIR449A in the anterior pituitary. MIR449 is induced by glucocorticoids and blocks the CRF signaling by suppressing the expression of CRF receptors and stopping the subsequent secretion of ACTH and cortisol and corticosterone. However, we found that the expression of MIR449A was not induced in low birth weight rats after stress exposure, and that this mechanism may be related to an increase in the expression level of Gas5 long non-coding RNA. Gas5 has a homologous sequence to the glucocorticoid responsive element, GRE. Gas5 acts as a GR decoy by having the same sequence as GRE and prevents GR binding to GRE by competitive inhibition. Therefore, in low birth weight rats, stress-induced corticosterone did not increase the expression of MIR449A, and the CRF receptor remained more highly expressed. Therefore, it appears that corticosterone is maintained at a high level in these rats. We recently found that among the representative GR-expressing tissues we examined, Gas5 expression was increased only in the pituitary gland. Unfortunately, the reason for this is still unclear. In a similar way, when comparing the response to glucocorticoids by the expression level of GR's co-chaperone factor, FKBP5 mRNA, we also found that glucocorticoid response was impaired only in the pituitary gland. We found that low birth weight rats had higher blood pressure than controls, and feeding a high-fat diet to the low birth weight rats further increased their blood pressure. The mechanism of increased blood pressure is thought to be decreased renal nephron count, arteriosclerosis, and the RAA system, but no morphological abnormalities in the kidney and aorta were found in our model rats. Aldosterone was also increased with a high-fat diet, but there was no statistical difference between low birth weight and normal birth weight. On the other hand, the blood corticosterone level of low birth weight rats was not different with a standard diet, but was significantly higher with a high-fat diet. The high-fat diet increased the expression of MIR449A in the pituitary for the control rats, but not for the low birth weight rats. Thus, in low birth weight rats, the induction of MIR449A expression was impaired, the blood corticosterone concentration then increased, corticosterone bound to the mineralocorticoid receptor, and then blood pressure increased by promoting the reabsorption of sodium in the kidney. From these results, it seems that the impaired expression of MIR449A occurs not only in physical and psychological stress, but also in metabolic stress. In the last part of this video, I would like to talk about the effects of nutritional interventions on low birth weight rats. We supplemented the diet with folic acid and vitamin B12 to normalize the glucocorticoid response in low birth weight rats. This fortified diet was prepared as in this previous paper, the high methylation diet. This diet was fed to lactating or third trimester mother rats or post-weaning offspring, as shown here. The result was that GAS5 recovered to the control level with the intervention during the first week after delivery or during the third trimester of pregnancy. However, the intervention was not effective if given for only 3 days after delivery or after weaning. When comparing blood corticosterone levels after exposure to restraint stress, the results were the same. Intervention was only effective during the first week after delivery or during the third trimester of pregnancy. Elevated glucocorticoid levels present with various symptoms other than hypertension, as evidenced by Cushing's syndrome. We are going to analyze other symptoms caused by the mismatch in our model rats in the near future. In summary, our model rats have a small body size caused by the trade-off of blocking the GH signal in the liver. It is thought that the mismatch between the phenotype and the high-fat diet environment caused an increase in blood pressure, like the Dutch famine type. In our model, abnormal glucocorticoid feedback is one of the subphenotypes found in the thrifty phenotype. Therefore, past epidemiological studies and the animal models we have established have shown the following in common. First, exposure to malnutrition during the embryonic period causes low birth weight and sometimes short body length and low body weight due to impaired catch-up growth. Second, these rats have an impaired negative feedback mechanism of glucocorticoids in the pituitary. Interventions with methyl modulators, such as folic acid and vitamin B12, have a critical window, but can partially normalize the response of blood corticosterone levels to stress exposure. Finally, these rats have elevated blood pressure regardless of compensatory growth. High glucocorticoid levels may be involved in this mechanism. In the future, we would like to clarify the following points. The first is identification of body composition determinants. We would like to clarify what factors and organ associations regulate body fat percentage and skeletal muscle mass. The second is elucidation of the acquisition mechanism of the thrifty phenotype. The third is proof of predisposition in low birth weight. We would like to identify the risk-forming factors for developing diseases. And finally, epidemiological studies and animal experiments have also shown that small physiques affect the next generation. So we would like to discover the generational propagation mechanism and develop an intervention method that can prevent these adverse effects.